Hello students, I am Dr. Kowsalya, Professor from the Department of Food Science and Nutrition, Avinashalingam University for Women. Can you reiterate the concept of genetic engineering we have seen in the previous class? Yes, genetic engineering is used for the production of valuable proteins, organic acids, enzymes, hormones in prokaryotes and others like flavor saver tomato, golden rice and the like in eukaryotes. So, how do you think this is possible? Can you guess? Yes, by use of certain enzymes. We are going to see today how the enzymes act as the major tools for in vitro manipulation of DNA and RNA. The term, as all of you know, in vitro means in test tube condition or in laboratory, whereas in vivo means inside the living organism. The various applications of molecular biology and the entire success of recombinant DNA technology is based on specific functions of these enzymes. Therefore, this lesson will introduce you to the various enzymes used in genetic engineering process. Coming to the objectives, after completing this lesson, you will be able to know the concept of various enzymes used in genetic engineering. To comprehend the steps involved in cleavage of vector DNA by restriction endonucleases, and to learn the use of ligase, which is an important enzyme, and also other enzymes in the formation of recombinant DNA molecules. To see the overview of today's lesson, this module is a part of the food biotechnology paper. One of the techniques involved in food biotechnology is the formation of recombinant DNA molecules by genetic engineering. A genetic engineer, what do you think about it? He is one who manipulates genes. The molecular toolkit of a genetic engineer is nothing but enzymes and cloning vectors. Recombinant DNA technology or genetic engineering play an important role in understanding the concept of genetic engineering. To see the overview of today's lesson, this module is a part of the food biotechnology paper. One of the techniques involved in food biotechnology is the formation of recombinant DNA molecules by genetic engineering. A genetic engineer is one who manipulates genes. The molecular kit of a genetic engineer is nothing but enzymes and cloning vectors. Recombinant DNA technology or genetic engineering plays an important role in understanding the concept of gene cloning. Thus, the enzymes are used in the formation of DNA library. In this module, you will be able to understand the actions of different enzymes used in genetic engineering process. Coming to enzymes as tools, what are enzymes? Basically, we know enzymes are protein catalysts and carry the forward reaction of a substrate. Enzymes involved in genetic engineering obviously act on DNA strands and show specific action. Let us see the enzymes that are used as tools of genetic engineering and gene cloning. What are nucleases? Nucleases, of course, are a category of enzymes that catalyze the hydrolysis of single nucleotide present in a chain of DNA. Nucleases play a vital role in analyzing the sequence of nucleotides in RNA and DNA. Nucleases are categorized as exonucleases and endonucleases. What are exonucleases? They are enzymes that act upon the genome and digest the base pairs on 5' or 3' ends of a single stranded DNA or at single strand nicks or gaps in double stranded DNA. Exonucleases are of two types. One is lambda exonuclease and the other is exonuclease 1. Lambda exonuclease enzyme removes nucleotides from 5' end of double stranded DNA. The mode of action of this enzyme is shown here. As you see here, an improved substrate for terminal transferase is formed. Therefore, this enzyme is used for 5' modification. Coming to exonuclease 1, the mechanism of action is shown here. As you see here, this enzyme removes nucleotides from 3' hydroxyl end of the double stranded DNA fragment. Consequently, an improved substrate is formed which is used in gene manipulation. This enzyme is used for 3' end modification of DNA fragment as shown here. What are endonucleases? They act upon genetic material and cleave the double stranded DNA at any point except the ends, but their action involves only one strand of the duplex. Hope you are able to catch up? Right. You must have heard about restriction endonucleases. What are they? They are called molecular scissors. They are the basis for recombinant DNA technology. These enzymes are present in bacteria and provide a defense mechanism called restriction modification system. The molecular basis of this enzyme was approached by Werner Haber in 1965. However, during the early 1950s, it was discovered that when phage DNA, you must have heard about restriction endonucleases. The restriction endonucleases are called molecular scissors. They are the basis for recombinant DNA technology. These enzymes are present in bacteria and provide a defense mechanism. You must have heard about restriction endonucleases. What are they? They are also called molecular scissors. Restriction endonucleases are the basis for recombinant DNA technology. These enzymes are present in bacteria and provide a defense mechanism called restriction modification system. The molecular basis of this was approached by Werner Haber in 1965. However, during the early 1950s, it was discovered that when lambda phage infects E. coli, phage DNA is delivered inside the cell. Host control restriction modification system provides defense to the E. coli. The bacterium protects itself through a process called restriction mechanism, which means that the E. coli identifies the introduced foreign DNA and cuts into pieces called restriction endonucleases and another process called modification system, which means methylation of certain bases of its own DNA, which is done by the enzyme methylase, so that the lambda phage expressed restriction endonuclease could not cleave its own DNA. As you see, this figure shows the restriction modification system. If phage is propagated on E. coli strain K, it is allowed to infect E. coli strain B when the efficiency of infection is very slow, whereas the progeny phage obtained from plagues of B strain can reinfect E. coli strain B with high efficiency. But the infection efficiency of these progeny phage on strain K is also very slow. There are three gene loci, HSDS, HSDM and HSDR, that control this system. The HSDS encodes a protein which governs the specificity of these systems. The HSDM gene product acts as modification enzyme, that is methylase, which interacts with HSDR gene product, that is restriction enzyme, in the cleavage process. It may be explained that the progeny phage particles first grown on strain K and then strain B are restricted when grown on B strain. Do you understand? But the progeny phage particles from strain B are not restricted when reinfect strain B. Again, they are restricted when grown on strain K. This shows that the efficiency with which phage grows on a specific host depends on the host strain on which it was propagated previously. Therefore, this confirms when the restricted phage is adsorbed on the surface of the restrictive host and injects its DNA, it is broken down quickly into restriction endonuclease. Hence, the restrictive host protects its own DNA from degradation and modifies its enzyme enzymatically. Thus, the restriction activity is provided by two enzymes. One is a nuclease and another is a methylase, the genes of which reside on the host chromosome, or you name it, a plasmid. What are the types of restriction endonucleases? Yes, there are three different types of restriction endonucleases: type 1, type 2 and type 3. Each enzyme differs slightly by different mode of action. Type 1 and type 2 are large multi-subunit complex which contains both endonuclease and methylase activity. Coming to type 1 restriction endonucleases, what are they? The BK restriction and modification enzyme systems on E. coli have been termed as type 1 endonucleases, such as EcoB and EcoK. They are complex nucleases which function simultaneously as an endonuclease and a methylase. They move along the DNA in a reaction and require magnesium, S-adenosyl methionine, which we say as SAM, and ATP as cofactors. They are single multifunctional enzymes consisting of three subunits. One is restrictive subunit, another is modification subunit, and the third one is specificity subunit. The third subunit specifies the recognition sites. The enzymes show specificity for recognition site but not for cleavage site. They recognize specific sites within the DNA but do not cleave at these sites. Hence, heterogeneous population of DNA fragments is produced. The recognition site of type 1 enzyme is 15 base pairs long and the cleavage site is about 1000 base pairs away from the 5' end of the recognition sequence, which is present within the 15 base pairs long recognition site. Coming to the important category, type 2 restriction endonucleases. These restriction enzymes were first isolated by Hamilton Smith. They are simple, consisting of single polypeptide chain and require no ATP for degradation of DNA. They have separate activities for cleaving DNA, that is endonuclease activity, and methylation, that is methylase activity. They are the most stable enzymes which require magnesium as cofactor. Daniel Nathans first used these enzymes for mapping and analyzing genes and genomes. The first type 2 restriction enzymes were isolated and characterized from E. coli strain namely RY. Hence, this was named as EcoR1. Do you understand? For the discovery of restriction enzymes, Arbour, Smith and Nathans were awarded with the Nobel Prize in 1978. Very interesting, isn't it? Out of the above three types of restriction endonucleases, type 2 are used in gene manipulation. Type 1 enzymes recognize a specific sequence of DNA molecule but cut elsewhere, whereas type 2 makes cuts within the restriction sites and produce two single strand breaks, one in each strand. Type 2 enzymes are the most important ones. Do you know how the nomenclature of restriction enzymes is done? Right. The restriction enzymes are named based on the principles, namely, number 1, the name of the organism that is identified by the first letter of the genus name and the first two letters of the species name to form a 3-letter abbreviation in the italic. For example, Escherichia coli. The first letter is E and the first two letters of the species is CO. So, you name it as ECO. Similarly, for Haemophilus influenza, it is HIN and it goes on like this. The second is a strain or type identified is written as a subscript. For example, ECO-K for E. coli strain K and HIND for Haemophilus influenza strain RD. The third one is in such cases where the restriction and modification systems are genetically specified either by a virus or by a plasmid, the extra chromosomal element is identified as a subscript. For example, you name it again as EcoR1, EcoP1, etc. The fourth one is that when a strain has several restriction and modification systems, these are identified by Roman numerals. For example, HIND1, HIND2, HIND3 for Haemophilus influenza strain RD and thereon. These Roman numerals should not be confused with those in the classification of restriction enzymes in type 1 per se. Do you catch up? Okay. Now, what are recognition sites? Restriction enzymes usually recognize a specific DNA sequence of 4, 5 or 6 nucleotides in length and cleave the DNA within the restriction site. There are 4 bases in DNA which are randomly distributed. The expected frequency of any particular sequence can be calculated as 4n, where n is the length of the recognition sequence. Thus, tetranucleotide sites will occur every 256 base pairs and pentanucleotide sites will occur every 1025 base pairs and hexanucleotide sites will occur every 4096 base pairs. The complementary sequences are also called palindrome sequences or palindromes. What are they? The recognition sites are the actual palindromes or palindromic sequences. The palindromes are the nucleotide pair sequences that are the same when you read forward, that is left to right, or backward, that is right to left, from a central axis of symmetry. That is, two strands are identical when both are used in the same polarity, that is in phi dash to 3 dash direction. For example, the phase shown here reads the same in either of the directions left to right or right to left. What about cleavage site? For example, the restriction endonuclease cuts between G and A residues of each strand and produce two single stranded complementary cut ends which are asymmetrical, having 5' overhangs of 4 nucleotides. These ends are called sticky ends or cohesive ends. Do you catch up? Okay. On the other hand, some other type 2 restriction enzymes cleave both ends of DNA at the same base pairs but in the center of the recognition sequence and results in DNA fragments with blunt ends or flush ends. For example, He3 isolated from Haemophilus aegypticus, the order of the enzyme 3, 4 nucleotide long palindromic sequence and cuts symmetrically both DNA strands and form blunt ends. Hope you are able to understand. Shall we see some examples of restriction enzymes with their recognition sites? As you see here, HAE3, which has a recognition site at GC or CG, it cuts the DNA strands and produces blunt ends, whereas the next enzyme, as you see here, BAMH1, which cuts at GG, produces cohesive ends or sticky ends. The next example is HIND3, which recognizes AA. Therefore, you get a cohesive or sticky end here. For example, again, in ECO-R1, the recognition site is GA or AG, where you are able to get a cohesive end or a sticky end. I think now you have understood very clearly the action of restriction endonuclease 2. What do you think are the importance of restriction enzymes? Restriction endonucleases are used for dissecting, analyzing and reconfiguring genetic information at molecular level. Restriction endonucleases are used in cleavage mapping, preparation of DNA probes and of course in gene cloning. What about type 3 restriction enzymes? The type 3 enzyme is made up of 2 subunits. One specifies for the site recognition and modification and the other for cleavage. In a reaction, it moves along the DNA and requires ATP as a source of energy and magnesium as cofactor. ATPase activity is lacking in these enzymes. Some examples of type 3 enzymes are HPA1, MBO2, FOK1 and the like. They have symmetrical recognition sites and cleave the DNA at specific non-palindromic sequences. For cleaving double-stranded DNA, 2 sites in opposite orientation must be present. One strand of double-stranded DNA is cleaved about 25 to 27 base pairs away from the recognition site, which is located in its immediate vicinity. Recognition sequence of some of the type 3 enzymes are shown here. Since the cleavage products of these enzymes are homogeneous population of DNA fragments, they cannot be used for genetic experiments. So far, we have seen independently the action of restriction endonuclease 1, 2 and 3. Now, you see here a comparative picture of their activity, nature, cofactors required and of course restriction and modification systems. You see here it is given clearly for type 1, type 2 and type 3. You must have heard about transcription, reversal of transcription. So, what do you understand from the enzyme reverse transcriptase? Reverse transcriptases are enzymes that are used to synthesize complementary DNA or copy DNA using mRNA as a template. To construct a cDNA clone bank and to synthesize cDNA molecules, reverse transcriptases are used. We know already that genetic information from DNA is passed to protein via mRNA as a mediator. During 1960s, Temin and co-workers identified that even informations can pass from RNA in certain experiments with cancer caused by animal viruses. This led to the discovery that DNA can be used as a template to synthesize copies of viral RNA in a cell. In 1970, Mizutari, Temin and Baltimore discovered thereby that informations can even pass from RNA to DNA. Retroviruses which possess RNA contain RNA-dependent DNA polymerase, which is also called as reverse transcriptase. Now do you understand why they have named it as reverse transcriptase? Okay. These results with the formation of single-stranded DNA that acts as a template for the production of complementary DNA. Let us see the general functions of reverse transcriptase. They are: 1. It acts as RNA-dependent DNA polymerase which transcribes single-stranded DNA from viral RNA. It demonstrates the activity of ribonuclease H, that is RNase H, which is a subunit of the reverse transcriptase enzyme. And 3. Reverse transcriptase act also as DNA dependent polymerase and transcribes the second DNA strand, which is complementary to its first DNA strand. Coming to the important enzyme, namely DNA ligase. What are they? They are also called DNA joining enzymes. DNA ligase forms a specific type of enzyme in molecular biology which facilitates the joining of DNA strands together by catalyzing the formation of a phosphodiester bond. Mertz and Davis in 1972 for the first time demonstrated that cohesive termini of cleaved DNA molecules could be covalently sealed with Ecoli Dna Ligase and were able to produce recombinant DNA molecules. It plays an important role in repairing single-stranded breaks in duplex DNA in living organisms, but some forms such as DNA ligase 4 may specifically repair double-strand breaks, that is, break in both complementary strands of DNA. Single-strand breaks are repaired by DNA ligase using the complementary strand of the duplex as a template with DNA ligase, creating the final phosphodiester bond to fully repair the DNA. The DNA ligase has applications in both DNA repair as well as DNA replication. In addition, DNA ligase has extensive use in molecular biology laboratory for recombinant DNA experiments. Purified DNA ligase is used in gene cloning to join molecules together to form a recombinant DNA. What is the mechanism of ligase action? Shall we see this? Okay. The mechanism of DNA ligase is to form two covalent phosphodiester bonds between 3' hydroxyl ends of one nucleotide which acts as an acceptor with a 5' phosphate end of another which acts as a donor. ATP is required for its action, which proceeds in 3 steps. One is adenylation, which is a lysine residue is adenylated in the active center of the enzyme, thereby pyrophosphate is released. Number 2, the transfer of the AMP to the 5' phosphate of the so-called donor, thereby in the 3rd step the formation of a phosphodiester bond between the 5' end of the donor and the 3' hydroxyl of the acceptor. Coming to the types of ligases, you may be wondering from where they are isolated. They were originally isolated from viruses. They also occur in E. coli and other eukaryotic cells. The types of ligases are Ecoli Dna Ligase, T4 DNA ligase, thermostable ligases and of course mammalian ligases too. Coming to Ecoli DNA ligase. The Ecoli DNA ligase is encoded by Lig gene. The DNA ligase in E. coli as well as most prokaryotes uses energy that is gained by cleaving nicotinamide adenine dinucleotide, that is NAD, all of you know, to create the phosphodiester bond. It does not ligate blunt-ended DNA except under conditions of molecular crowding with polyethylene glycol, and they cannot join RNA to DNA efficiently. Coming to the important, most commonly used enzyme, namely T4 DNA ligase, which is isolated from bacteriophage T4. It can ligate cohesive or sticky ends of DNA, oligonucleotides as well as RNA and RNA-DNA hybrids, but not single-stranded nucleic acids. It can also ligate blunt-ended DNA with much greater efficiency than Ecoli Dna Ligase. Unlike Ecoli Dna Ligase, T4 ligase cannot utilize NAD and uses only ATP as a cofactor. And some engineering has been done to improve the in vitro activity of T4 DNA ligase. Coming to the mammalian ligases, in mammals there are 4 specific types of ligases: DNA ligase 1, DNA ligase 2, DNA ligase 3 and DNA ligase 4. The DNA ligase from eukaryotes and some microbes uses ATP rather than NAD. Coming to the 4th category of thermostable ligases, they are from various thermophilic bacteria which have been cloned, sequenced and are available commercially for use in amplification reactions because of their thermostable properties. What is homopolymer tailing? The complementary DNA strands can be joined together by a process called annealing. This principle is utilized in homopolymer tailing. The technique involves the addition of oligo-DA to 3' ends of some DNA molecules and the addition of oligo-DT to 3' ends of other molecules. The homopolymer extensions, by adding 10 to 40 residues, can be synthesized by using terminal deoxynucleotidyl transferase from calf thymus. Homopolymer tailing achieved by annealing is shown here. I think you are able to understand. Coming to linkers and adapters, how are they used? Linkers and adapters are chemically synthesized short double-stranded DNA molecules. Linkers possess restriction enzyme cleavage sites. They can be ligated to blunt ends of any DNA molecule and cut with specific restriction enzymes to produce DNA fragments with sticky ends. Adapters can contain performed sticky or cohesive ends. They are useful to be ligated to DNA fragments with blunt ends. The DNA fragments held to linkers or adapters are finally ligated to vector DNA molecules. Coming to alkaline phosphatase. What is the role of alkaline phosphatase in genetic engineering? It is an enzyme which is involved in the removal of phosphate groups. This enzyme prevents the unwanted ligation of DNA molecules, which is a problem usually faced in cloning experiments. When the linear plasmid DNA is treated with alkaline phosphatase, the 5' terminal phosphate is removed. This prevents both recircularization of plasmid DNA dimer formation. Now, it is possible to insert the foreign DNA using DNA ligase. Thus, the sticky end of one vector DNA and sticky end of DNA insert on ligation produces recombinant DNA molecule. What is the role of alkaline phosphatase? Alkaline phosphatase is an enzyme which is involved in the removal of phosphate groups. This enzyme prevents the unwanted ligation of DNA molecules, which is a problem usually in cloning experiments. When the linear plasmid DNA is treated with alkaline phosphatase, the 5' termini phosphate is removed. This prevents both recircularization and plasmid DNA dimer formation. Now, it is possible to insert the foreign DNA using DNA ligase. Thus, the sticky end of one vector and the sticky end of another DNA insert on ligation produces a recombinant DNA molecule. Polymerases are another category of enzymes used in genetic engineering. What are they? They are a group of enzymes that catalyze the formation of nucleic acid molecule. Polymerase enzyme act on nucleic acid template. There are three important polymerases. 1. DNA dependent DNA polymerase, RNA dependent DNA polymerase, and the 3 DNA dependent RNA polymerase, which produces RNA from DNA. Another enzyme is SI nuclease. Have you heard about it? It degrades the single stranded DNA or single strand protrusion of double stranded DNA with cohesive ends. As a result of action of S1 nuclease, cohesive ends are converted into blunt ends. Therefore, S1 nuclease is used to remove the incompatible ends so that overlapping ends may be developed for annealing of two fragments of DNA molecules. To conclude, students, in this module, you have learnt the various enzymes used in genetic engineering. To summarize today's lesson, type 2 restriction endonucleases are used extensively in the field of recombinant DNA technology, and we have seen how the reverse transcriptase is used in cDNA synthesis and alkaline phosphatase to prevent recircularization of plasmid DNA. Further, ligation, a process involved in recombinant DNA molecules by ligases, is also discussed. Therefore, this module would help you to understand the process of gene cloning, which we are going to see in the subsequent lessons. See you in the next class.